All right, this is more on inheritance. We're going to go over the laws that Mendel discovered, talk about alleles and their behavior, and then start working with some simple genetics problems with Punnett squares and pedigrees. And so the illustration here just shows Mendel's uh, the basics of Mendel's experiment where he crossed true breeding purple flowered peas with true breeding white flowered peas. And he found that all the offspring were purple flowered. But then when he allowed these to self-fertilize, he found that 3/4 of the individuals were purple flowered and 1/4 were white flowered, so that the trait that was recessive reappeared in the second generation. So that was Mendel's basic kind of the most important discovery he had was that there could be dominant and recessive alleles, that recessive alleles could remain hidden and then show up in subsequent generations where heterozygotes were allowed to self-fertilize and they showed up in a very specific ratio. Um, this and Mendel did this with 7 different traits, and he showed that these traits didn't affect each other, that they were all inherited independently. Um, and Mendel, from there he concluded that traits were inherited in discrete packets. There was no blending of information, that individuals had 2 packets or 2 alleles of that they received from their parents, one from each parent for every trait, and these could come in a dominant or a recessive form. That when individuals made their gametes, they would pass one of those alleles onto their offspring, and which one they passed on was entirely at random, and that the inheritance of his trait, this one trait, didn't affect other traits. And so Mendel was able to determine this because he could control the fertilization of the plants. He had carefully selected the plants and knew that they had these discrete traits that he could follow. And then he would find true breeding varieties and cross them together. And Mendel did an enormous amount of collecting data with large numbers of offspring. And the reason he was able to do this was because he had a background in agriculture and he knew selective breeding and he knew how it worked. Um, One of the things we haven't talked about, but they talk about in your book, is that Mendel kept track of 2 traits at the same time. And so he referred to this as a dihybrid cross. And so here, like for seed color and texture, he kept track of the 2 different traits at the same time and found that they were inherited independently based on chance. And instead of the 3-to-1 ratio, he got offspring in a 9-3 to 3-to-1 ratio, where 9 out of 16 individuals showed the dominant trait for both traits. 3 individuals showed the dominant trait for seed color and the recessive trait for seed coat. 3 showed the dominant trait for seed coat and the recessive trait for seed color. And 1 out of 16 showed both the recessive traits for seed coat and seed color. Okay, this picture shown here is quite confusing. It's been the bane of genetics students for decades. And this is just a fancy version of a Punnett square where we follow 2 traits. We're never going to make you do that in for a grade, but I will use Punnett squares, and you will have to do a Punnett square problem on the next exam and in lab. And those Punnett square problems are just ways of keeping track of the types and proportions of gametes that the parents can produce and the types and proportions of offspring. So it's used to predict both the genotype and the phenotype of offspring. Uh, we start with a box with 4 squares for when we're talking about a single trait. On the left side of that, we put the 2 alleles that the mother has. On the top, we put the 2 alleles that the father has, and then we we just carry down and across to see what the offspring should have. Um, and so here we have for eye color, both the mother and father are heterozygotes, having one gene for brown eyes and one gene for blue eyes. Okay, and here we see one of the offspring will have two dominant genes. Two of the offspring will have one dominant and one recessive gene, and one of the offspring will have two recessives. And so this gives us the ratios of 1/4 being homozygous dominant, 1/4 being homozygous recessive, and 2/4 or one half being heterozygotes. Okay, both the heterozygotes and the homozygous dominant will have brown eye color. So the genotypes will be different from the phenotypes. Okay, here we're going to do a problem kind of together. Here, this is about an autosomal recessive disease. Steve and Linda have a history of cystic fibrosis in their families. Steve is a carrier and Linda of cystic fibrosis, and Linda is a heterozygote. They always wanted to have a family with 3 children. Okay, complete a pedigree showing these 5 people, the parents and the 3 children. Complete a Punnett square for Steve and Linda. So there's the Punnett square. Cystic fibrosis is shown as the small h gene. Both Steve and Linda are carriers. Both are heterozygotes. Okay, their offspring are shown right there. Okay, uh, 1/4 of the offspring will have 2 dominant genes and be normal. 2/4 will have one dominant and one recessive and will be a carrier, and 1/4 will be homozygous recessive and be affected by cystic fibrosis. Okay, so any one child has a 3/4 chance of being healthy. Okay, and so each individual child has a 3/4 chance. Here we show the child is 2 boys and 1 daughter, and each one is 3/4 healthy. What is the probability of having one healthy child? It's 3/4. And what is the probability that all 3 children will be healthy? It's 20. You just multiply 3 times 3 times 3 times over 4 times 4 times 4, and that's 27/64. So something like 1/3 or so is the chance that all 3 children will be healthy. So likely that they might have 1 or 2 children with the disease. Okay, here's another one with hemoglobin. This is a system where there's 3 different alleles, um, or excuse me, 2 different alleles, the normal hemoglobin and the sickle cell hemoglobin. Homozygous individuals, if you have 2 dominant genes, you're susceptible to malaria. If you have 2 recessive genes, you'll have sickle cell anemia. The heterozygotes are resistant to malaria. And so sickle cell anemia is an autosomal recessive disorder. Jane is normal and does not carry the gene, whereas her husband does have the disease. What percentage of their children will have sickle cell anemia? Okay, well, Jane is normal. She has 2 copies of the dominant gene. Her husband has 2 copies of the recessive gene. He has sickle cell anemia. And so their children will both inherit dominant genes from Jane. And so all of their children and will have one normal gene. So none of them will have the disease, but both children will also inherit the recessive gene from their father. So they will all be carriers. Okay, and so this is typical of a recessive gene. And in this case, we'll often skip generations because you can have 2 normal parents and be a carrier. Again, here you'll be normal. Um, you'll only produce affected children if you marry another carrier, and so then you can produce children that are affected. Here, in generation 3, we see a carrier marrying another carrier. This is one of the reasons why it's a bad idea to marry your cousins, because if you do carry recessive genes, it's likely to come out when you marry a related individual. And so don't do that. Okay, so we're going to practice this more in lab. Mendel he outlined the basic methods of inheritance, and he did this by controlled breeding of very familiar plants. He kept records over multiple generations and followed simple, discrete traits where he could identify recessive and dominant. He used very large samples and used probability to explain his results. He determined that organisms carry two alleles for each trait. One allele was inherited from each parent. Alleles could be dominant or recessive, and the genotype may differ from phenotype. For the traits Mendel studied, they assort independently, but we know that's not always the case. We've used Mendel's work to unravel more advanced understandings of genetics. And so here's a picture of a mother and an albino child. The mother is obviously a carrier of the gene for albinism. The child's father is also a carrier. So we'll stop there.